Hello to our friends joining us via recording. We are wrapping up our discussion of lesson number 11, which is about the brain. When we were last in class together, we were talking about the reticular activating system, or RAS for short. Remember that the reticular activating system is what helps you to stay alert and oriented, as they say in the medical field, or essentially what keeps you conscious. So when we're thinking about what helps you to stay conscious, it's things like visual inputs. So when you see that there's sunlight outside, that helps you know to stay awake. When you hear things, so auditory information, like the sound of your alarm clock going off to wake you up. Or when you feel things like temperature or pain or touch sensations. All of these feed into the reticular activating system to tell it to send out little pings or little messages to your entire cerebrum to keep you awake. I don't know if we talked about this entirely. When it's time for us to go to sleep, we turn the reticular activating system off temporarily. But did we talk about what it would be called if the reticular activating system gets damaged or it's deactivated in a way that it shouldn't be? What happens if the reticular activating system doesn't work? Did anyone get it? I like it. Hayden even found us a GIF for it. Absolutely right. When the reticular activating system gets damaged to the point where it can't turn back on, a person is in a coma. Now, an important idea, because later in the packet, when you're wrapping it up from your studying, it'll ask you, hey, if a person's in a coma, what is and isn't working? The critical thing when we're considering that is the job of the reticular activating system is to keep the cerebrum awake. So a lot of the other more basic parts of the brain that are doing their basic jobs, I'm talking things like your breathing rate or your heart rate. Those kinds of things could still be happening in the background. This is why someone can be in a coma and not need a ventilator. Their body may still be alive, but their mind may not have any activity. So the reticular activating system specifically activates the cerebrum. It doesn't play a big role in some of those more basic life functions. And I believe the last thing we chatted about on Monday was the fact that there's one kind of sensory information that does not feed into the reticular activating system. Do we remember from our class on Monday which kind of sensory information is missing from here? Yeah, exactly. We're starting to chime in. The kind of information that does not feed into the reticular activating system to keep you conscious and awake is olfactory information. So this mattered for us when we were talking about a house being on fire. When your house is on fire, if you're awake, you would smell that first. But if you're asleep, without having some kind of auditory impulse or some kind of visual impulse, you may not wake up. At least you may not wake up until it's really hot or you're being hurt by a fire. So we use smoke detectors to help us kickstart our reticular activating system so we can get out of the house in time. Let's talk about the opposite of the reticular activating system, and that is a structure we talked about in lab called the pineal gland. So the pineal gland gets its name because it's shaped like a pine cone. Remember in the sheep brain that this was a dark spot? The pineal gland makes the hormone melatonin, which is the sleepy time hormone. Now, normally our body is supposed to make melatonin more when we get close to bedtime and less as it's time to wake up. But I think we can all relate to our friend here in the picture up here because it's that time of the semester when we just have melatonin all the time because we're exhausted. In addition to exhaustion, maybe throwing off our melatonin secretion, we can also experience problems with melatonin related to jet lag. So if your pineal gland is spitting out melatonin like it would have when you were in a different time zone, that can make you tired at the wrong time of day. So the way that we can address jet lag is exposing you to what's called bright light therapy. So if you see my picture here, literally this woman is sitting next to a very bright light, and it makes her truly happy. Look at how happy she is. So you sit next to a bright light, you take in that high energy wavelengths of light like would come from the sun, and that trains your pineal gland. Now is not the time to spit out melatonin. I need to wait. So that's why often when you're traveling over many different time zones, they'll recommend that you actually stay up until bedtime in your new time zone to help your pineal gland get synced with it. So jet lag, an example of an issue with the pineal gland. And hey, as a sidebar, I'll mention, you've probably heard people ranting about how bad blue light is for you. They're actually not wrong. They might be a little bit misguided and a little bit over the top about it, but blue light is light that has the highest amount of energy. So we'll talk about that a little bit in the special senses chapter. Back in caveman days, when we didn't have smartphones and those kinds of things, the only time we saw blue light of that much energy was from the sun. So our body is adapted to, whenever we experience that kind of wavelength of light, to think that it's time to stay awake. So before bedtime, when we're scrolling through our phones or watching TV, we're sending mixed signals to our brain and our pineal gland, because we're giving it the kind of light exposure that it would have seen in the daylight, it would have seen with the sun. So that's kind of where that idea comes from, of issues with screens before bedtime. Yeah, so Lisa mentioned that there are glasses. Absolutely, there are blue light blocking glasses to help those wavelengths of light not be perceived in your eyes as so energetic, to help offset some of that glare or that kind of feedback into your body. The pineal gland, if you remember in your lab packet, there was a term that I told you to kind of ignore, the epithalamus. That's what the pineal gland is part of, the epithalamus. So for lecture, we're just talking about the pineal gland, and lab, we're pretty much just talking about that as well. So from the epithalamus, I'm going to take you into the thalamus. When we talk about the thalamus in the brain, consider this to be a relay center. So information comes into the thalamus, and the thalamus figures out which direction in the brain it needs to go to. So if I'm bringing in sensory information related to temperature, I might send that here, versus if I've got some sight information coming in, I might send that here, or hearing information. The thalamus helps to sort information to the right place of the brain for it to be processed. But the other thing that the thalamus does is it filters that information. So some of the information that comes into your mind, you will not remember it. You'll never consciously perceive it, because the thalamus says, yeah, that's not important. I'm not going to let them know that. So a good example of this, since we're having an online class together, would be the fact that right now, you may not realize you're wearing pants. I mean, some of us might not be wearing pants, right? Some of us might be in leggings or shorts or pajamas. We're online, right? Anything goes. But Dr. Allis is wearing pants, for the record. I am at school in my pants. But when I'm wearing any kind of clothing, or when we're in the classroom together, I say when we're wearing shoes, you don't notice that you're wearing them unless you start to consciously think about it. Because your thalamus says, okay, I felt this sensation before, I don't care about it anymore, I'm done. So when we think about the thalamus, it sends information where it needs to go, and it also edits that information. Things that you don't need, it's going to chop out, you'll never perceive them, and it will allow you to focus on other kinds of things. So I'm popping back up those fMRIs that we looked at in class together on Monday, reminding us again, right, that it takes way more brain space to think than to speak. But what I also want to emphasize here with the thinking fMRI is the fact that so many different parts of the brain are all working at the same time. The thalamus is helping us to direct information from the prefrontal cortex, where we're figuring things out, to some of our memory centers, to what we're hearing, When we think, that thalamus helps to bridge a lot of different places in the brain to help them to work together. So that's one of my functions of the thalamus. If we move down into the brain stem, we're getting into the part that, if you remember from Monday's quiz, was the part that was basic life functions. So the brain stem is what helps us with things like our breathing rate. It's what helps us with things like our heart rate. So primarily breathing rate and heart rate are the pons and the medulla. You'll talk about that a lot more in AMP2, but the pons and the medulla work together to determine how quickly you're breathing, how deeply you're breathing. The other thing that the medulla helps us out with is with lots of different reflexes. So these are reflexes for things like vomiting, sneezing, or coughing, all of those things done by the medulla. So the medulla oblongata, very involved in reflexes, as well as the heart rate and the breathing rate. We mentioned at the beginning, remember, if someone's in a coma, these parts of the brain may still be working. And in fact, unless these were damaged, they probably are still working. But the functions of these parts of the brain are not thinking, feeling, emotions. It's basic life functions, what keeps your body physically alive. So keep that in mind with the brainstem. When we talk about keeping your body physically alive, one of the most important things that we do for it is give it lots and lots of blood. Every time your heart beats, we send about 20% of all of the blood that leaves your heart up to your brain. So compared to its size, the brain takes way more blood than the rest of the body. There are two things that we find inside blood that the brain needs a lot of. Can you help me out in the chat? What are the two things inside the brain or in the blood that the brain needs a lot of? Any guesses? Yeah, so lots of us honing in on the first big one, being oxygen. We definitely need oxygen everywhere in our body, but especially in our neurons. And the other thing that comes with the blood, in addition to that oxygen, is the nutrients. And I've seen a couple of us get specific, because we did talk about way back in unit number 1, how the nutrient that your neurons need to thrive is glucose. That's their favorite kind of food. That's their only kind of food. So we send a lot of blood up to the brain to make sure it has enough oxygen and to make sure that it has enough glucose. When we're thinking about someone having a stroke, what happens in a stroke is that we have a blood clot, for example, right here. And a blood clot means we block the flow of blood from here forward. We cannot survive very long. Our neurons can't survive very long without fresh oxygen and nutrients. This is why if someone has a heart attack, you immediately start CPR, because the longer the brain goes without oxygen or nutrients, the more those neurons stop being able to function, the more they start to die. So we send a lot of blood up to our brain to make sure that that our brain has all of the oxygen and the nutrients that it needs. Most of the blood vessels in the brain are covered by what we call the blood-brain barrier. I'm typing that in the chat, the blood-brain barrier. The blood-brain barrier is essentially two sets of plasma membranes that are found between the blood vessels and the tissue of the brain. So the first set of membranes comes from the blood vessels themselves. And we actually talked about this kind of blood vessel, again, way back in unit number 1. What was the name of the kind of blood vessels that do not leak? The kind of blood vessels that protect me from viruses or bacteria leaving the bloodstream to get into my brain tissue. Yeah, exactly. It was mean for me to ask you to type that much, right? We talked about the kind of blood vessels called continuous capillaries. So continuous capillaries, the entire plasma membrane of the, or excuse me, the entire wall of the blood vessel is completely solid. So it's made out of cell membranes and there are no holes in between. Nothing's going to leak. But when we talk about the blood-brain barrier, the other thing that goes along with that is this orange line that we see right here. And this orange line is actually another layer of cells. These cells are called astrocytes. They get their name because they're shaped like stars. So astrocytes wrap around on the outside of the continuous capillaries, and they provide another layer of filtration. So I've got two complete plasma membranes between the blood and the brain to help it to stay safe, to help things not get through. I am promising you that on the exam, I'm going to ask you a question about what could or could not cross the blood-brain barrier. The answer to that question is going to be exactly the same as the answers to the question from unit number 1 when I said, hey, what can cross the plasma membrane of a cell? Because all the blood-brain barrier is 2 cell plasma membranes. So the kinds of things that can cross the membrane of a cell can also cross the blood-brain barrier. Things like oxygen or things like carbon dioxide. When we were talking about those molecules, would we say that those molecules were polar or non-polar? Things like oxygen or carbon dioxide, polar or non-polar? Yeah, several of us agreeing in the chat. Oxygen, carbon dioxide, these were examples of non-polar things, things that share their electrons equally, so they have no charge. Just like those things can sneak across the plasma membrane of one cell, they can sneak inside and outside the blood vessels as well. So non-polar things, or here's another way of saying the same thing, fat-soluble things, things that would mix with the tails of those membranes, they can go straight through as well. So anything non-polar, anything fat-soluble or lipid-soluble, that can go straight out of the blood-brain barrier. Sometimes I need things to get across that actually are polar. Things like glucose, for example. Glucose, my favorite food. I'm going to have to do transport to make that happen. And when I'm talking about transport, I'm talking about using proteins to move things across the membrane. So I do glucose transport, where I use proteins to sneak this polar thing from one fluid into my brain, where my cells are that need it. I'd have to do the same thing with a lot of my amino acids, except for the non-polar ones. Everybody else is going to have to go through a protein tube. But there are some things that I do not let across the blood-brain barrier. One of those things are things with charge. I do not let negatives or positives get across. Anions or cations, they're stuck because they have a charge. And you know what else I don't let get across the blood-brain barrier are things that are large. So any kind of protein, any kind of molecule that's big is not going to be able to get across the blood-brain barrier. And that can be problematic if someone happens to have an infection in their nervous tissue. I can't just give them oral antibiotics in their mouth and expect that to go into their bloodstream and get to their brain, because the blood-brain barrier says, yeah, no, you're too big. I'm not letting you out. So with some kinds of infections, we actually have to give that medication into the subarachnoid space, the place where that cerebrospinal fluid is. If I need something to actually get to the brain tissue, I have to find a different way to get it there, because the blood-brain barrier blocks everything. I don't care if you're a good guy or a bad guy. Yeah, Lisa was mentioning we don't let white blood cells through. We also don't let antibodies through. So if there's an infection, our brain is on its own, which is why we have that blood-brain barrier to keep it safe. There is one place, though, in the brain that I do not have the blood-brain barrier, and that is a place that we talked about during our lab class last week when we talked about the choroid plexus. So the choroid plexus, when you're thinking of these are the leaky blood vessels that we have in the brain. I need them to leak because they are the source of cerebrospinal fluid. So cerebrospinal fluid is the fluid that's inside our ventricles of the brain. I make cerebrospinal fluid by leaking plasma out of the blood vessels. My ependymal cells from lab, they filter it, and as long as there's nothing bad in it, they'll release it into the ventricles as cerebrospinal fluid. Even though we've got these filtering cells here, it's important to say that if you were to get a brain infection, 100% confident or 99% confident that this would be the place that it came from, because these blood vessels are already leaky. They're already letting stuff out. The ependymal cells will do their best to filter it and make sure that nothing bad comes out, but the blood vessels are leaky. So if I got an infection in my nervous tissue, this is probably where it came from the choroid plexus. Yeah, so Lisa mentioned an example of something that we could have in the brain called meningitis. Yep, meningitis is when those meninges that we talked about, the connective tissue around the brain, when those get inflamed, that's problematic. Yes, so meningitis, an example of a disease that affects technically the connective tissue around the brain, but it puts extreme pressure on the brain, definitely. We have choroid plexus in all of the ventricles. So in the lateral ventricles, in the third ventricle, even down in the fourth ventricle. I'm making cerebrospinal fluid every single day in each of the ventricles. And if I'm going to be making it all the time, I also need to be dumping it all the time, which is where those things we talked about in lab, the arachnoid granulations, that's where they come into play, allowing us to release that cerebrospinal fluid into the dural venous sinus, where I can recycle it back into the bloodstream. And now that we've all had lab this week, remember that we also talked about that central canal this week too. So we send that cerebrospinal fluid down into the spinal cord as well. But we can have issues with the fluid getting stuck in one of the ventricles, or the fluid not being able to drain into the dural venous sinus. And when that happens, that's called hydrocephalus. I'm going to type that in the chat, hydrocephalus. The word means water on the brain, but it's not a water problem. This is an issue with cerebrospinal fluid. So if for some reason one of my passageways gets blocked in the ventricular system. So remember, I'm going to bounce back here. Remember we talked about in lab how I could block the interventricular foramen. Then I'm going to need to build up a fluid in here. Or I could block the cerebral aqueduct. My fluid could build up in here. If I can't drain that fluid and I keep making that fluid, I end up with an abundance of that fluid in and around my brain. In infants and in toddlers, hydrocephalus typically would manifest itself kind of like this, where we see the skull expanding outward. But when we talk about hydrocephalus in adults, hydrocephalus in adults can very quickly be fatal. There's something that babies and toddlers have that adults do not have. What do we have in babies and toddlers that makes hydrocephalus a little less fatal? We'll still definitely treat it, but it's less likely to be fatal. Yeah, exactly. Remember that when we talked about a fetal skull or a young baby's skull, they have those soft spots called fontanelles. And the fontanelles are the protein layers that go between the bones. Remember that the fontanelles do allow movement. They do allow stretching. So the skull bones have actually expanded out because they can. But in the adult skull, instead of having fontanelles, we all have sutures, which means that if we get extra fluid and that fluid's inside the ventricles and starts pressing on the nervous tissue, our brain doesn't have anywhere to expand. Our skull bones are set. So what ends up making it potentially fatal is this dramatic increase in intracranial pressure. So the pressure that's being put on your brain, or more specifically, on your brain stem. Can you remind me in the chat, what did we say that the job of the brain stem was? So things like the pons and the medulla. What's the job of that part of my brain? Yeah, exactly. These are my basic life-sustaining functions. This is breathing, this is heart rate, this is the normal stuff I need to stay alive. If I have too much pressure down there and they stop working, that's absolutely fatal. So the brain stem, very important for keeping you alive. We obviously want our cerebrum to not be in too much pressure either. But if the brain stem gets under too much pressure as well, that's when we'll see things like the heart stop beating or breathing rate go sporadic, those kinds of things. To treat hydrocephalus in adults, and sometimes actually in children as well, we'll put in a thing called a shunt. So it's a tube that goes from the skull. Typically we'll embed it right underneath the skin in the neck, and that tube will run down through the chest into the abdominal pelvic cavity and actually drain that fluid consistently from the ventricle down into that cavity where it just is in the body system. So if there's some kind of obstruction, that's one way that we can continue to drain that fluid, because remember, we're always making it. If we're not always dumping it, we're in trouble. That is all I've got from lesson number 11. Before I turn off the recording, any last minute questions about brain stuff? Or shoot me a GIF or an emoji. How do we feel about the brain? I like it. Yep. Max and I went the same direction. My brain's dancing. Lisa's brain is meh. Yeah, so Jamel asked a follow-up question about how a shunt would put that fluid into the cavity. The insides of our body are not dry to begin with. They have some level of lubrication in general. So I would presume that with the cerebrospinal fluid going in there, it just kind of gets added to the mess that is inside of you. We can actually reabsorb some of the fluids depending on the kind of I just lost my train. Oh, the kind of tissue that it's going on top of. So that would be a possibility as well. Jessica's mentioning that she thought that shunts drain into the bladder. That could be possible. I'm not familiar with that to say either way. To my knowledge, they typically just drain into the cavity, but I could be incorrect because it's been a little while since I've read up on my shunts. But I can confidently tell you that we embed them under that skin in the neck to get all the way down into either the abdominal pelvic cavity or the bladder, whichever the correct answer is on that. All right, well, I'm going to stop the recording and let me make sure. I just saw something come in. Lisa asked a follow-up question on the CSF if staying awake causes a buildup of it. There is actually a very interesting article for Current Events in Anatomy about how when you're sleeping, your brain actually goes through predictable patterns of essentially washing itself with cerebrospinal fluid. So I don't know that it necessarily builds up the cerebrospinal fluid, but that article goes into how they're talking about it with neural diseases. So brain diseases, if we're not essentially washing the brain every night like we do when we're sleeping, that can lead to problems like Alzheimer's, where we build up proteins that are non-functional that the CSF might have helped to wash away. So that article, if you're interested in reading about that, yeah, we have predictable patterns that we cycle that cerebrospinal fluid through while we're sleeping to help wash our brain. Great question.